Hi everyone, here's my first lecture video for Bio 1C. We'll see how this goes in the future, how much we'll do of the pre-recorded stuff and how much we'll do of it in person. But for now, we'll start talking about the cell cycle and cancer. And so I definitely want to discuss a lot of this with you all in person because it's really interesting and relevant and certainly affects a lot of people and their health. So first, to think about cancer, you have to understand the cell cycle of cells. And so we'll go through a little bit of basics here before we talk about it in class tomorrow. And so what we're going to talk about for this topic is going to be genetic material, so to think about DNA and what it is, how it's distributed, cell division, so mitosis, and this will include your cell division lab for Friday. And then we'll talk about regulation of cell division, and then finally cancer, which really is going to be dysregulated cell division. So we got to know about the regulation piece first before we can talk about what it looks like when it's dysregulated. Okay, and so as a review, your genetic material, which I'll move my little talking head down here. Okay, so the genetic material is a substance that contains the information for how to build an organism. And so how our cells know what to do, the instructions that they use to do what they do is going to be DNA, right? And so all living animals and plants and organisms on this planet are going to be DNA based. We have some RNA based things like viruses, right? But most things, the instructions are going to be DNA. So the instructions in your cells, the DNA, tell your cells what to do. And we'll get to this topic later on in the class, but your DNA genome holds the information to make every protein in your cell, right? So this is the process of gene expression, is reading that genetic material, reading that DNA, and making proteins from it. And so this whole process includes transcription and translation, which we'll get into detail later on in the course. Okay, so because all of your cells have the same DNA, you have to replicate that DNA because you need to go from one single fertilized egg cell into a 30 trillion celled person like you are today. So each time that a cell divides, it has to replicate its DNA. Genetic material is also inherited, right? So if you are making new organisms or thinking about yourself, right? You inherited genetic material from the egg and sperm that made you. So you have to think about replicating DNA and inheriting that DNA. All right, so first we got to think about how DNA is organized. So DNA is organized into chromosomes, and a chromosome is not just the DNA itself, it's actually the DNA wrapped around these proteins called histones, okay? So when you take this long blue strand here, that's the double helix, you wrap it around these proteins called histones, and then you kind of pack those little strings together, okay? And what this gives you then is this compact DNA that's relatively organized, right? So if you actually look at DNA under a microscope, look at chromosomes, which we'll do in some labs coming up, we'll see the chromosomes. It's this packaged DNA here, okay?
I think I got kicked out of.
Okay, sorry for that. I was talking about how chromosomes are packaged into chromatin. And so this allows this chromatin to organize and compact the DNA. So if you really took out a DNA strand and strung it out the whole length, it would be way, way, way bigger than the cell. So to get all of those strands packaged into the cell, you have to organize it into chromatin. And what this ends up doing is it actually allows us to regulate DNA. So what happens is that when the DNA is condensed, okay, and the chromatin is condensed, the genes can't be expressed. When it's looser, they can be expressed. And so we'll talk about that process a lot more later on too, but just know for now that this condensed chromatin allows the cell to regulate what DNA is expressing at any given time. So when you look at chromosomes, often you're going to see this little pinched section in the middle. That's called the centromere. And this becomes really important during cell division when cells are organizing where chromosomes go, okay? So the centromere in the middle of each chromosome, and it's going to be in a different spot depending on which chromosome you're talking about. Okay, and so remind you that a gene is a unit of DNA sequence that codes for a specific protein, and those proteins are contributing to traits. So we're not talking about one gene for a trait, we're talking about one gene for a protein, and multiple proteins contribute to a trait. Thinking about your flies, for example, a white-eyed fly might have a white protein, but it might be multiple proteins that contribute to eye color. Okay. Okay. So how DNA is organized into chromosomes. In prokaryotes like bacteria, you have a single circular chromosome. In eukaryotes like us and plants and animals, we have linear pieces of chromosomes. Okay. So here's how it would look in a plant, an African blood lily. You can see this condensation here of chromosomes. And when you lay them out like this in pairs, that's called a karyotype. Okay. All right. So what a genome is the entirety of an organism's hereditary information. Okay. So in E. coli, if you laid out the DNA lengthwise, it would be about 0.006 feet. If you laid out our DNA, it would be about 6 feet, okay, per cell. So your genome is the entirety of an organism's hereditary information. And your genome then is all your information, and this provides the instructions for cell growth, maintenance, protein expression, all this fun stuff, right? And what I want you to think about before we talk tomorrow is, do you think that genome size correlates with organismal complexity? Okay. So what do you think? Think about it at home. Maybe don't go to the next slide yet, okay? But think about this, and then tomorrow in lecture, we'll talk about this, okay? So I will see you all tomorrow for more discussion of cell division. Think in the meantime whether you think genome size would correlate with organismal complexity. Do you think that more complex individuals would have bigger genomes?